whatever.
It was just a fault of the finger.
I was traumatized. I was like 9.
When Hey, I'm gonna wake you in about half an hour, okay?
Now, okay, we are live, as they say, and today we're going to take a look. We're going to start chapter 3. This is unit 2. We're going to look at the cells, and in the second part of unit 2, we're going to take a look at metabolic activity. And so this should take us a couple weeks to get through here. The lecture test, I will open up the lecture test next week. I'll let you know. Probably going to have the lecture exam in class next Thursday, and that's when the online test will open up too. So you can, that way we'll get at least 2 good lectures in, or 2 lectures. I don't know how good they're going to be, but we'll get 2 lectures in. So let me go ahead and send this around and okay. Anybody have any questions on anything relevant or irrelevant? You know, so I'm perfectly amenable. So, okay, then I'm going to go, would the practice test be advisable? The practice test is the question is, of course, is the practice test advisable to study for the lecture test? Practice test will give you an idea of what to expect. The questions on the practice test are not identical, they're similar. My practice tests were old lecture exams, probably from 8 or 10 years ago. And there's only so many ways to ask the same kind of question. I would review the PowerPoints first, because that's where I draw the questions from. If I didn't mention it, then it's likely if I didn't mention it, it's not going to be on the test. I'll just put it like that. But then you could test yourself. You could question what you have retained by taking the practice test, but don't depend entirely on the practice test. If you have to make a choice, review the PowerPoints first. Thank you. Any other questions? Exactly, all the PowerPoints, everything we've done for chapter 1 and chapter 2. And I will tell you that chapter 2 will have probably more questions on it than chapter 1. There'll probably be more about the chemistry, but not much more, because I try very hard to give equal weight to the first chapter, as boring as it is. So, okay, well, now the good news is we're done with chemistry, and we'll never see chemistry again after that test coming up. So now let's take a look at something a little more biological, and that is cells. Now, first of all, let's say, every living thing is made of cells. Only living structures have cells. Every living organism, whether it's a bacteria or a fungus or a fish, a dog, a cat, a human, a plant, a tree, they're all made of cells. Viruses are not living, so viruses are not made of cells. Viruses are only a strand of DNA surrounded by a coat of protein. So when they always say on the news, well, we're going to kill the virus, okay, it's already dead. But it's easy to say we're going to kill the virus and we're going to destroy the viral structure, by breaking apart its protein code. So anyway, organisms are made up of cells. There is nothing smaller than a cell that's alive. So the basic unit of life is a cell. There is no something smaller than a cell. And cells only come from other living cells. There's no way to create artificial life. We can't mix together ammonia and methane and oxygen and carbon and hit it with electricity and have something alive. It doesn't work that way. Cells only come from other living cells. Now in our bodies, we have 200 different types of cells, and they are all different sizes and shapes. The longest cells are the nerve cells, and the nerve cells run the length of every nerve they're in. So the longest nerve in our body, the sciatic nerve, which starts at our hip and ends up at our big toe, those nerve cells are probably 3 or 4 feet long, depending on your stature. Some are very small. Red blood cells, for example, very tiny. White cells, very tiny cells. Different cells have different shapes and different structures for what they're going to do. So muscle cells are long. They run the length of every muscle they're in. So if you're looking at your biceps, for example, if your biceps is 4 inches long, all the cells in the biceps are 4 inches long. There are no little cells and big cells. Every muscle cell will run the length of the muscle it's in too. So different sizes, different shapes. But regardless of the type of cell, it's going to have 3 things in common with all other cells. It's going to have a membrane. It's going to have a plasma membrane. That's the outer covering of the cell. Plasma membrane is like the skin. It keeps things out and keeps things in. It's very protective. Our skin is like that too. So it doesn't matter what kind of cell it is, whether it's a bacteria or a fungus or us, it's going to have a membrane. Even plant cells that have a cell wall have a membrane underneath the cell wall. The cytoplasm is the goop or cell sap, I once heard it called that, that's inside, that fills up the inside of the cell. It's the liquid, semi-liquid gel makeup that all the organelles, the nucleus and all the other little structures that work for us float in. It's mostly salt water. It can be goopy. It can be pretty liquid. It just depends. And then the third thing that every cell has is a nucleus. The nucleus is where the DNA is located. All of our DNA is found in the nucleus of every cell, and the DNA is contained there. It doesn't leave the nucleus. It sends instructions out to the cell using the other nucleic acid, the RNA we talked about. Now, the only cells that don't have DNA in them don't have a nucleus, and those are red blood cells. Red blood cells have kicked their nucleus. They had a nucleus when they were being developed, but they kick it out so they can use that space for carrying more hemoglobin. In fact, they don't have anything in there. Red blood cells are just a big old sack of cell membrane filled with hemoglobin to carry oxygen. That's all they do. They don't reproduce. They don't do anything else. They just carry oxygen. But when they were forming in our bone marrow from the blood stem cells, they had a nucleus up almost to the very end of their formative process. Then they kick it out. So we can still say that all cells have or had a nucleus. So there's what a typical cell might look like. Cells are surrounded by a layer of water. Our bodies love water. We use water for all sorts of things, and we have what we call the interstitial fluid that surrounds the outside of the cells, and it's salt water. And the salt water outside the cell is the same concentration as the salt water inside the cells. So the cells are filled with water, and we have water surrounding them. That water, that interstitial fluid, is nothing more than plasma that has leaked out of our vessels. Because our blood vessels have lots of openings in them. They're not a solid, inflexible tube. They've got lots of openings and clefts and holes that plasma leaks out. When it leaks out, it becomes interstitial fluid and it surrounds the cells. And then we recover it as lymph and return it back to the plasma. So it moves around a lot. Now the structures that we see here, the big purple thing in the middle is the nucleus. And that's where our DNA is located in the form of chromatin. And we have lots of tubules, flattened tubes that surround the nucleus all the way around. And we've got lots of smaller little organelles, like those big orange things that look like sausage links, are the mitochondria. And mitochondria is where we break down sugars to release energy. Every textbook ever written refers to mitochondria as the power plant of the cell. And I try to avoid saying that because I don't like the phrase. So all the mitochondria does is takes, which is what they do, it's pretty amazing. It takes sugar and burns that sugar to release all the energy trapped in the covalent bonds. So it's a little more than just a power plant. You know, power plant, it sounds like making a power plant makes it sound like it's a little simple operation, and it's not, it's pretty complicated. We also see that we have all sorts of projections on the surface of our cytoplasm. We've got numerous organelles inside the cell to do all sorts of things. We make proteins, we package them up, we ship them out, we release energy, we prepare for mitosis, we generate all the different proteins we need, we engulf foreign substances or food, we do all that, and every cell, except like the red cells, have that capability. So the cell membrane is a phospholipid bilayer. It's made up of phospholipids, a double layer of phospholipids. The phospholipids look, their phospholipids are based off of a triglyceride. If you remember, I remember to press the right button, there we go. Okay, here's your triglyceride. You have carbon, carbon, and carbon. This is glycerol here. It's an alcohol, and you have the fatty acid tails here. A phospholipid, instead of having 3 fatty acids, it has a phosphate group attached over here, and it has 2 fatty acid tails. And so the phospholipid doesn't have that 3rd fatty acid chain. Furthermore, the chain is bent. So it is a unsaturated fatty acid. Now, why is this important? Well, it looks something, let's see if I can bring that, get rid of all of this and go to the next slide. This is what the phospholipid bilayer looks like. They set themselves up in a double layer with the phosphate heads setting out, pointed out to the outside and pointed to the inside of the cell. So for example, this is the outside here. Here's the inside of the cell. And each one of these is a phosphate. And these are the fatty acid tails here. Now the fatty acid tails are not bound together with each other with van der Waals. They're loose, which lets them have great flexibility. The whole surface of the cell membrane is like a big squishy bounce house, if you will, or imagine a great big old water balloon. It's all squishy, maybe a water balloon filled with oil. I don't know, but it's very, very squishy, and it doesn't have a lot of structure to it. It'll move up and down, and things in the membrane will slide across. And let's see if I can show you what this is saying here, and hopefully we can hear it.
Is selectively permeable. It lets some substances through. The plasma membrane is selectively permeable. It lets some substances through and not others. The hydrophobic interior of the bilayer is one reason membranes are selectively permeable. Hydrophobic molecules, those that are soluble in lipids, can easily pass through the membrane. In addition, small molecules like O2 can sneak between the phospholipids of the membrane. On the other hand, large hydrophilic molecules like glucose and ions, such as sodium ions and hydrogen ions, cannot pass through the membrane unaided.
So this phospholipid bilayer, for whatever reason, the membrane sets itself up in this double layer, and it's based off of fats. I mean, the phospholipids are fats, and so anything fat soluble, like the cholesterol-based hormones, for example, the sex hormones and the adrenal gland hormones, can pass right through. Anything that dissolves in fat can go through these membranes. Vitamins that are fat soluble, for example, like vitamin A and E and vitamin D, they slide right through. The ones that are water soluble have a hard time getting through. They don't get in because oils and waters don't mix well together. Look at you have Italian dressing. You have to shake it up, right? Because the oil sits on top of the liquid portion is mostly vinegar, and you have water, and the oil contains, and you have all the seasonings throughout, and the oil goes to the surface, to the top of it, and the water stays below. And that's because they're, as I say, it's not miscible. They won't mix together. That's my big word for the day, miscible. And so water doesn't get through. How do we get water into the cells? Well, there is a workaround in here. There are proteins that go through the cell membrane, and we call them channel proteins. And the channel proteins are just a hole, an opening, and water can go through this way. It can bypass the restrictions. Now oxygen, as I said, is so tiny, it just slides on through, it doesn't care. And CO2 can go out the other way, because it's also very tiny. Water can't get into the cell. Now, and water can't leave the cell. But if we have channel proteins, then they can go back and forth. Now, whatever leaves the cell is usually matched by whatever's coming in. And so we will have a movement of water back and forth, but it's usually a balanced movement. If you have ions that you want to get, how about glucose? Glucose is a nice big molecule, H12O6. It's too big to get through the membrane, but if we use another type of protein called a carrier protein, it will allow glucose to come into the cell. This is what insulin does for us when we eat carbohydrates. To get the glucose into our cells, to get the sugar into the cell that we need to do things, insulin allows the cell to make more carrier proteins, so we bring more glucose into the cell, more sugar into the cell for energy. And once we, and when we bring our blood sugar level back to where it belongs, our insulin works, our blood sugar goes up, we eat something, we eat a donut, we eat an apple, we eat a potato, we eat fries, we eat cereal, whatever, anything with carbohydrates in there. It raises our blood sugar. It goes above our set point value for sugar. And so we release insulin to let the cells suck up the extra sugar. When we do that, it brings our blood sugar back down. And we use the sugar then to do cell things. We power our cells and whatever they're going to do. Once we bring our blood sugar back down, we stop releasing insulin and we stop making these carrier proteins. So this is what the phospholipid looks like. It's a much better drawing than mine. The phospholipid, the big round pink structure, is the phosphate. I don't know if phosphates are pink, but in this case they are. We've taken off the third fatty acid chain to make a phospholipid. We attach the phosphate in place of that. That leaves you the two fatty acid tails. Well, one of those tails is unsaturated, so it has a kink in it. And so they, like you would see in a liquid fat, like in oils, the phospholipid layer is squishy, if you will. It's like a squishy, semi-liquid layer, because they're not stuck together. If they were van der Waals, if they were held together with van der Waals attraction, our cell membranes would be rigid. It'd be almost like our own cell wall, but we don't get that. We get these squishy little tails that move all around. They're all flexible because our cell membranes actually do move up and down, side to side and up and down. They're very, very flexible. And there's another picture of the phospholipid bilayer. And it really does look like this. So now this is probably a more detailed drawing of what our cell membrane looks like. The whole surface of our cell is covered with this phospholipid bilayer, which is constantly in motion. It's moving up and down, and all those structures in blue can be sliding around in the membrane. They're not locked in, most of them are not locked into a specific place. It's like if you were to take a water balloon and roll it in cooking oil and then roll it in Lucky Charms. And I picked Lucky Charms because it has all the different shapes in there. And as the balloon moves, as the water in the balloon moves, the Lucky Charms floating on the oil are going to move around too. And that's sort of what's happening on our cells, because the surface is in constant motion. All these structures in here are moving around all the time. Now what we see here, we see proteins. These things in blue are what we call we have what's called come on, you were just there. There you go. Okay. Here is what's known as an integral protein. Integral proteins go all the way through from one side to the other. That could be a carrier protein. It could be a channel protein. Here is a peripheral protein on the side, here on the inside. Peripheral proteins usually only show up on one side of the membrane. They do all sorts of things, like function like enzymes or identifiers, or they help with the channels or the carrier proteins. The carrier proteins and the channel proteins are going to go all the way through from one side to the other. That's why they're called integral. They extend all the way through the membrane. We have markers, those little green things and blue things sticking up that look like little branches coming up. Those are markers telling us what the cell is, who it is. It also identifies certain regions in the cell where other molecules can react with it. We've got cholesterol embedded in the membrane. Cholesterol functions as like rebar in concrete. Rebar you put rebar in concrete. It's steel rods to reinforce the concrete. And that's what the cholesterol does here for us. As little yellow linked ring structures are the cholesterol, and the I guess I've covered all of that. So and we keep the phosphate heads facing outward and facing inward, so that not only can water and large molecules come into the cell, water and large molecules can't leave the cell unless they go through one of those specialized proteins. Get out here now. Okay. So the phospholipid bilayer is, the cell membrane is mostly phospholipids. 5% glycolipids. A glycolipid is a fat with sugar attached to it. It's a sugar-coated fat. Think funnel cake, you know. You sell pretzels, right? You don't sell funnel cake. So, but if you sell funnel cake, it's like sugar-fried fat, right? You know, I'm not dissing funnel cake. You know, it's got that melt in your mouth goodness, right? You know, so we're gonna just, you know. Anyway, but that's what the glycolipid is. It's a fat with a layer of sugar on top of it. Sometimes these glycolipids are used as markers to tell, you know, proteins that float throughout our bodies, hey, you can land here. You know, we use proteins as hormones. Hormones get released from our brain and tell other cells to do things. You know, like thyroid hormone comes out of our thyroid gland. Clever how that's the same name. Thyroid hormone comes out of the thyroid gland and tells the cells how rapidly they need to burn sugar. Thyroid hormone controls our metabolism. You talk about someone that has a high metabolic rate, it's because their thyroid is very active, or a low metabolic rate, their thyroid is not very active. But every cell in our bodies has a receptor just for thyroid hormone. And so maybe those glycolipids are functioning as the receptor to tell the hormone, here's where you land. So, and here's where you come into the cell. Other types, other combinations of glucose and protein are used as identifiers to say, I'm me, don't hurt me. And then there's, of course, there's a lot of cholesterol embedded in the membrane to give the membrane its structural strength. Now the proteins that stick through here, they're either going to be an integral protein or a peripheral protein. I know it's a highly detailed animation there. It's the best I could find. So, but what it's trying to show is that the membrane is always in motion. So the proteins are moving around too, but an integral protein will go all the way through the membrane. And it's a way to communicate, either to allow substances to go through the protein as a carrier protein or a channel protein, or to communicate with other cells. And so if it's an integral protein, it goes all the way through. It's a peripheral protein. It's sort of butted up against the inside of the cell membrane. And peripheral proteins have a big role to play in communicating within the cell. So the integral proteins function as transport proteins. They can be a carrier protein. This is where we get into that facilitating diffusion that we talked a little bit in lab. A carrier protein will change its shape to allow an ion or another molecule to come through at that site. And they can be real selective. They can let in glucose, they can let in sodium, they can let in potassium. But if it lets in sodium, it won't let in potassium. They're real picky about what they let in too. So those are carrier proteins. Channel proteins, water and maybe amino acids and stuff like that can slide right through. These integral proteins also function as enzymes. They can have chemical reactions taking place on the surface of the cell. So we do a lot with these proteins. Now the peripheral proteins can also be enzymes, but they can also be the connections between the cells. They can also be the proteins that cause muscle contractions. When we have cytokinesis taking place in the at the end of mitosis, cytokinesis occurs because there's a band of protein that forms around the cytoplasm and starts squeezing. It can contract, and this is why we call it a contractile protein. We use that same type of protein in our arms and legs and our voluntary muscles to cause our muscles to contract, to do things. So we have all sorts of functions here of these integral and peripheral proteins. And if you really want to read up on them, you've got lots of stuff to read here about transport or enzymes or receptors or joinings or attachments or recognition. Let's show you a couple of these here that are probably the most important ones.
Transport proteins carry molecules from place to place. The example shown here allows certain solute molecules to enter the cell. Hemoglobin is the transport protein that carries oxygen in the blood.
So in this case, the transport protein is a channel protein or a carrier protein that's going to open up and let sodium come in, or potassium come in, or calcium to come in, or potassium to go out. It just changes its shape. It's totally passive. This is some of that facilitated diffusion where the cell needs help to get something into or out of it. The other type of transport protein is hemoglobin. The example there, hemoglobin is a huge molecule. It's 360 amino acids in length, but it packs into all the red cells, and we use that as the way we carry oxygen from location to location in our blood. That's all hemoglobin does. Now, the second type here.
Receptor molecules bind to signal molecules and can then emit second messengers which trigger changes inside a cell. Receptors are thus important links in the system of communication among cells. Some signal molecules, such as hormones, are also proteins.
Now these hormones are called chemical messengers, and we've heard of some of them. We've heard of growth hormone probably, and thyroid hormone. I just talked about that. Some of these hormones are so large they can't go into the cell, so they have to land on the surface of the cell on a receptor, which then tells an adjacent protein, go tell the inside of the cell that we're here and do what we're supposed to do. You know, because just because it's a great big hormone doesn't mean it's going to get in. You know, if it's not fat soluble, it can't get in. Growth hormone is very large. It has to depend on a second messenger. That's what that little structure is, a little green structure that goes, it's turned on inside the membrane. And we use those mechanisms all the time. Now this is structure.
Structural proteins have many functions. Like tent poles and ropes, they shape cells and anchor cell parts. They may serve as tracks along which cell parts can move. They bind cells together, making organized units such as muscles, ligaments, and the tendons that bind muscles to bones. The silk of spiders and the hair of mammals are also structural proteins.
So our structural proteins hold us together. You know, like the collagen fibers that attach to that are wrapped around our muscles that attach to bones. Without them, we couldn't move. Because our muscles alone aren't strong enough to do what we want to do. We have to pull on bones and use the bones as levers to get things done. So all these structural, these shapes that our cells have and the things that we do depend on these structural proteins. You know, flagella and cilia depend on the proteins to allow them to move inside the cells or on the outside of the cells. And this last category that we see are the enzymes.
An enzyme is a protein that changes the rate of a chemical reaction without itself being changed into a different molecule in the process. Enzymes promote and regulate virtually all chemical reactions in cells.
You know, we remember with the chemical reaction, we can't do anything about the temperature because chemical reactions occur faster at high temperatures. We can't change the temperature in the body. They occur faster when you increase the reactants, not the amount of reactants. We're stuck with microscopic amounts of reactants, and we're stuck with very small particle sizes. So we can't alter anything, any of those conditions. What we can do is make an enzyme that will bring the reactants, the small number of reactants we have, it'll bring them together quickly. And so the one thing we can, by flooding the area with the enzyme, is the one thing we can do to make reactions go without a lot of extra energy. And so we have enzymes inside the cell, outside the cell, on the surface of the cell to control all these reactions. Now, here is a very important structure, and we'll come back to this several times, particularly in AP2. This is the glycocalyx. This is the carbohydrate, the sugar that can be attached to a protein or to a fat. It's a glycolipid or a glycoprotein. The glycocalyx structure is a marker telling us who we are. Every cell in our body is going to have glycocalyx structures that stick up and identify us as self. Now, when we are about 1 year old, all of our white blood cells, what we call the T cells, go into, half of all of our lymphocytes, the large white blood cells, go to our thymus gland and become trained to identify all the glycocalyx structures in our bodies. They become immunocompetent. They can recognize self. They recognize us as, who we are. And so they don't attack them because they're part of white blood cells. The T cells are part of our immune system. But you get a foreign cell in there, a foreign bacteria, a virus, which will still also have a glycocalyx on there. You get a transplanted tissue, you get a mismatch on blood transfusion, and the immune system will respond to it and attack it and try to destroy it, or at least reject it or whatever it does. And so the glycocalyx structure tells us who we are. We call these glycocalyx structures our own antigens. We call them our antigens. And the antigens are found on the surface of every cell. And antigen. And our body's immune system creates antibodies that will block or destroy or target for destruction the antigens. So the glycocalyx structure is essentially our antigens. Every cell in our body has a glycocalyx structure that's recognized. Our blood cells all have identical markers on them, depending on your blood type. If you are type A, then everybody that has type A blood has the same antigen, the same glycocalyx on our red cell, in addition to the cells marking it as us. If you're type B, you have the same markers. Everybody has the same markers. If you're A, B, or O, they're all identical. So we separate out the antigens based on blood type. They tell us our blood type. And we'll talk about that in AP2. It works out very well for us. We run into problems when we have transplants. A person who has a transplant has to take some sort of immunosuppressant drug to suppress the immune system so the immune system doesn't attack the transplanted tissue. Or when we do blood transfusions, we have to make sure it's the right blood type. So, because giving someone the wrong kind of blood has some potentially fatal consequences. So it's not something that you just, it doesn't matter, give them whatever you want. It's red, it's not going to work. So, and the glycocalyx structures are very, very tiny. What we're looking here, microvilli that you see on the illustration here, microvilli, those are those folds of tissues down there that look, that look like a fence post, like a row of a fence along there. Those are folds in the cytoplasm of the cell membrane that are sticking up off of larger folds. The cell will have a fold called a villi. Villi means finger. Microvilli are little folds coming off of the villi. So this is microscopically small. And the glycocalyx structures are even smaller than that. So, but we have lots and lots and lots of these markers there. Yeah. Problems with the glycocalyx, besides, of course, the whole issue with transplants and transfusions and things like this, is that cancer cells don't play by the rules. Cancer cells have never played by the rules. Cancer cells will, first of all, they don't believe in contact inhibition, where they will, contact inhibition says cells will not grow over top of each other. It's like that each cell has to have its own personal space, even though they're butted up against each other. There's not another cell on top of it. So humans practice contact inhibition all the time. I mean, most of us are all sort of scattered apart here. Even before we had social distancing, nobody would sit on top of each other. What happens in cancer cells is they grow on top of each other. That's the first thing that cancer cells do wrong. The second thing is that they keep changing their glycocalyx, so they can keep passing as normal cells. It takes our immune system about 14 days to recognize a foreign antigen as foreign and respond to it. It just takes that long to produce antibodies to it. But somewhere along the line, right before we get to our 14-day recognition day, the cancer cell changes its antibodies, and it keeps doing that and keeps doing that. So our own immune system, which would normally protect us from things, can't respond. It can't react to the cancer cell because it doesn't see it. It will be, our immune system will be triggered by the foreign antigen, but by the time we mount a response to it, it has changed. So the cells that it would destroy aren't there. As far as they don't see them because, well, where'd they go? The antigen's different now, and then it to start over, and then it keeps going on like that. Now, something else we see about our cells is that they communicate to each other. They don't operate independently. They communicate to each other all the time. They're held together to each other. We have 3 different ways that cells join together. We have what are known as tight junctions. We have desmosomes, and we have gap junctions. Now, a tight junction is what we have where we don't want to have anything, where we want to have 2 cells held together, butted up against each other with a connection so tight that nothing can get through. So we have our stomach. Our stomach contains hydrochloric acid. The cells making up the lining of our stomach are held together with tight junctions. And so hydrochloric acid, the stomach acid, can't leak through into our abdominal cavity. That and the fact that we coat the inside of the stomach with a very thick alkaline mucus to neutralize the acid, but we can't get anything to leak through. So we have these tight junctions. We have blood vessels in our brain that are wrapped by glial cells, the same type of glial cells you saw in lab. The glial cells are wrapped around the capillaries in our brain and held together with tight junctions so that only certain things can come out of the blood into the brain cells. It protects the brain cells from things, substances that shouldn't be there. So tight junctions, it's almost like they are stitched together. The proteins within the membrane of each cell fuse together with the adjacent cell membrane. So it's like you've had something like it's been stitched, if you will. Because like on our clothes, where the seams are in our clothes, stuff can't get through. Now, desmosomes are a little different. Desmosomes are cells that have long rods of protein that go from one side of a cell membrane to another side, to the adjacent cell membrane, and then they're anchored off. They look like a zipper. They're very tight. They hold that cell together extremely tightly. It's extremely tight. They're anchored on both sides, and those blue things you see in the middle there are the proteins that are functioning like rods. Now, what this does is it gives you a great deal of strength, but a lot of flexibility. Our heart cells are held together with desmosomes because our heart has to contract every 0.7 of a second for 70, 80, 90, 100 years or more. And every muscle cell in our heart has to contract to its fullest extent every time. Every cell contracts. Every cell contracts as hard as it can. And so you want to hold those cells together, but you also want to have flexibility. And desmosomes let them have that flexibility because of that zipper-like structure that has a little give to it. It's still held, the cells are held perfectly together, but there's some flexibility there. And the third type are the gap junctions. Gap junctions are holes in the membrane that line up with each other. They're just like little holes or gaps, if you will. And the cells use, adjacent cells use those gaps to send nerve signals or chemical signals, but mostly nerve signals from cell to cell. In our heart, we use gap junctions to communicate because, you know, normally the way a cell works is an electrical signal comes to the top of the cell and has to work its way down into the cell and then go into the next cell and sort of hops along like that. But in the heart, by having these holes in the membrane, it just shoots right through here. And the heart uses it because, again, we have to contract every 7/10 of a second, and that is an electrical stimulus for that. And so we use these gap junctions as a quick way to communicate from cell to cell to cell. We see these in our muscle, in our bone cells. The osteocytes trapped in those little lacunae in there they have extensions that come out of each bone cell that will connect to an adjacent bone cell in another lacunae, a little bubble of bone in the bone, and they will communicate through gap junctions with those extensions of their cell membrane. So I don't want to get too excited about gap junctions. It's just, you know, that was sarcasm. You can laugh. So anyway, movement across our membranes depends on, it's either a passive process or an active process, and cells love passive. Who doesn't like something for free? You know, it's like, you know, you buy one get one free type of deal. We all like that kind of stuff. Cells are even better. They want free everything, and so they have either passive movement across the membrane or active movement. Well, the passive movement involves diffusion. Simple diffusion, you know, high concentration to a low concentration. Cells love that. They like to have facilitated diffusion, where you have some mechanism to help carry something across the membrane. It's still free. If you have a channel protein or a carrier protein, it simply changes its shape. When a molecule lands on it, it doesn't cost any energy to do that, and it makes things go into the cell or out of the cell. And then, of course, there's osmosis, the movement of water across the membrane from what we call high water to low water. We also filter. Our kidneys do an awful lot of filtering to clean out our blood. The capillaries filter all the time too, you know, and that's also passive. Somebody else has paid the price for the movement or the pressure or whatever. So cells love all sorts of passive processes. Simple diffusion does a great job for us with oxygen and carbon dioxide. Oxygen always comes across the membrane. Oxygen is always higher in the blood than inside the cell. So it will always go from the high concentration to the low concentration inside the cell. CO2 is always higher in the cell, and it will always diffuse outward freely, passively. Anything that's fat soluble slides right across the membrane. Cholesterol, steroid-based hormones, the sex hormones, for example, they slide right across the membrane with no problems whatsoever. Simple diffusion, just from the high side to the low side. This is simple diffusion right here. We put a dye pellet into this beaker of water, and you can see how it starts, you can see where the high concentration levels are, but gradually it will move from the high water, from the high concentration to the lower concentration until we reach equilibrium. And it's all passive, caused by the motion of the water molecules. Water molecules will always be randomly in motion. Water molecules don't stop moving until they're frozen. So the water molecules are moving around there and spreading the dipole around. The same thing would happen in here with the air molecules. If I opened up a container of banana oil. Banana oil is a chemical that smells like bananas. It's used for artificial flavoring, to give you that banana flavor. And so if I were to open it up and leave the container open, the molecules would gradually, banana oil would spread around the room because the air molecules in here are constantly in motion. Again, that's passive diffusion from the high concentration to their low concentration, down their concentration gradient. Now, diffusion is usually controlled by size of the particle, the temperature. If we were to turn up the heat, the odor would spread faster because the air molecules would move faster in the room. That's how turning up the heat works. You heat up the air molecules, and the air molecules hit your body at a higher rate. And the friction, believe it or not, the friction of the air molecule hitting our skin is what warms us up. So you may have noticed that we have heat in here today, as opposed to earlier in the week when it was, subarctic conditions. When I went down to see Dean Sutton and my nose was running and my hands were blue, she sort of got the idea that it was cold in here on Tuesday. So anyway, it's simple movement of the air. So it's all passive. This is how cells move things around, move from the inside to the outside, or the outside to the inside. It's all going to be a passive process, simply from the high side, the high concentration to the low concentration. CO2, always higher in the cell than outside. CO2 is always leaving the cell. Oxygen always comes into the cell. Now, if you are have a plasma membrane in place, it does add some complications. The plasma membrane is described as selectively permeable. Circle that, because that's a term you'll probably see again, selectively permeable. That's the classic description of the cell membrane, as a selectively permeable barrier. Meaning it lets some things go through and others not. Things that are water soluble don't go through on their own. Things that are fat soluble do. Things that are very tiny, like oxygen and CO2, can go back and forth across the membrane. They can squeeze through the gaps between the fat molecules, and the lipid membrane doesn't care. So if you're still depending on diffusion, you have to use some sort of mechanism, like a carrier protein or a channel protein. We have facilitated diffusion to get across the membrane. If you have a patient who has experienced a great deal of trauma to their skin, you run the risk of, from like severe burns, you're going to run the risk that your patient is going to lose too many fluids, too many ions, and not die necessarily from the burns, but die from water loss or some sort of infection that would take advantage of this burn condition. The cell, a person who's been severely burned, usually burned down to the bone. The most graphic analogy to think of is, consider what barbecued meat looks like, like barbecued ribs. That's what skin would look like that's burned to the 3rd degree. And so imagine being burned to the bone. The cells are damaged and destroyed. The cell membranes are ruptured. There are no ways to control water coming out of the cell. Your patients with severe burns will leak water rapidly. There's a tremendous amount of water loss from these burns. It doesn't hurt, usually. because all the pain receptors have been burned away. It'll hurt at the edges. But they're going to lose a lot of water, and they're also creating an environment where bacteria can grow. And so not only do they have tremendous amount of tissue damage, they're prone to some sort of infection, which may or may not kill them anyway. It's hard to control that water loss and the loss of the sodiums and potassiums in here when you have that much damage to the cell membrane. There's facilitated diffusion. I digress there in the burn there. Facilitated diffusion is just the use of the carrier protein or the channel protein. Here we see a carrier protein that can open and close. And the presence of the right ion or the right molecule that lands on it, because these carrier proteins are going to be selective. Depending on what the ion is or the molecule is, if it has a receptor site on the carrier protein, and all the carrier proteins are going to be different. If it lands on the right receptor site, it will change the shape of the carrier protein so that it can go on through. You'll notice how the substance lands on the top of the protein and it goes in. The bottom is now open to the inside of the cell and the top is closed until something else comes along. And then whatever that something else is, it lets it through. It might be sodium, it might be potassium, it might be glucose, it might be sodium that triggers glucose to come in with it. The carrier proteins are very, very selective. And usually, you don't have multiple ions coming in through the carrier proteins. A carrier protein for sodium is different from the one for potassium. So they can come in, they can go out. It just depends. And it's still passive. And then we have this thing called carrier-mediated facilitated diffusion. Here we're going to bring up across big molecules like glucose, or the amino acids, anything way too big for any other kind of protein, too big for an ion channel, a large molecule. This is what insulin does for the cells. Insulin allows them to make these large carrier proteins for, as we say, carrier-mediated facilitated diffusion. Glucose can't get into the cell any other way. Sugars can't get in any other way except using these specialized proteins. And here's some examples here. We have two types of facilitated diffusion. Ones that are always open, we call those leakage channels. Like, for example, on the animation there, you can see it. There's a potassium channel that's always open for potassium. That's always open. We call it a leakage channel. There is a gated channel. The gated channel controls access to the inside of the cell or to the outside through the use of a specific type of structure called a gate. And the gate can either be electrically controlled or chemically controlled. So if it's electrical, it's going to have to use energy from somewhere. But a chemical gate simply depends on the presence of something landing on it to cause it to open up. So leakage channels and gated channels. And of course, the water channels, which are nothing more than leakage channels, the water channels are called aquaporins. So, and they're only going to let water to come in. So everything up to this point has been passive. The diffusion, osmosis, facilitated diffusion are all passive. Everything going from its concentration down its concentration gradient. Osmosis is water going from high water to low water across the membrane. Diffusion is simply moving down from down its gradient from a high concentration to a low concentration, and the facilitated is still going from a high concentration to a low concentration, but this time we have help to get through. Active transport, on the other hand, requires energy. Cells sometimes need to get substances inside or outside that are moving against their concentration gradient. It's like they're pumping things, they're moving things uphill. Active transport uses up ATP. 40% of the energy the cell uses on a daily, on its being a cell is spent with active transport. Now, I mentioned osmosis already. Movement of active transport slide is probably out of place, but that's okay. Osmosis is not active transport. You're going from high water to low water across a membrane. It's still a free ride. The water can go through the aquaporins here, but we always move from the high water side of the membrane to the low water side of the membrane. It's the, depending on the water concentration. If, for example, the one here, if you have a 10% solution of sodium chloride, it's 90% water, because 10% and 90% is 100%. But a 40% solution of sodium chloride is only 60% water. Whatever the, the solute, the concentration of the solute determines the water concentration. 10% sodium chloride is 90% water. 40% sodium chloride is 60% water. When you have a membrane in the middle, you're always going to go from the high water side to the low water side. Like here's our membrane right here. Here's our 10% sodium chloride here, which is 90% water. Over here is 40% sodium chloride and 60% water, because both of those equal 100%. Now, which side of the membrane has the highest water concentration? The 90% side, right? Everybody, this is the high water because 90 is greater than 60. This is the low water. Water then moves from the high side to the low side to reach equilibrium. So water will move across a membrane into a cell from the high water side to the low water side. Now, what possible relevance does this have to humans? Well, consider this. You have a person who decides that they're really, really thirsty. And they got a whole case of water. They got a bargain at Walmart and got 60 bottles of water for $1. And they're going to start drinking one. They want to, it's sort of like, here, hold my beer and I'll watch me drink all this water type of thing. And so they drink one bottle after another, after another, after another. And pretty soon they've consumed about 6 liters of water in the space of 20 minutes. Now, where's that water all go? Where's everything we eat and drink go? Yeah, but first it goes into our blood. Everything gets filtered through our kidneys, but everything ends up initially in our blood. Everything we eat, everything we drink. So your tea and your coffee, and whatever you've got in there, I won't ask. But whatever you're eating and drinking ends up in our blood. Now, that 6 liters of water goes into the blood, and from the blood, a fair amount of it leaks out into the interstitial fluid. Now, that water is pure water, and it dilutes the interstitial fluid that surrounds the cells. Interstitial fluid normally is 0.9% sodium chloride, the same concentration as our plasma. But now you just flooded it with 6 liters of pure water. And your water concentration in the interstitial fluid around the cells has suddenly gone way up. You've diluted the salt. So what's going to happen to that water? It's going to move into the cells. What's going to happen to our cells? They're going to swell up and maybe pop. Blood cells can pop if they get if the fluid around them gets too dilute, because they're going to want to suck in the water. Cells will suck in way too much water. If there's too much water outside the cell membrane, the cell's going to try and suck it up to reach equilibrium. Because remember, water always goes from the high side to the low side. So a person who drinks a lot of water quickly can overhydrate themselves to the point that it becomes very, very dangerous. You know, people that run marathons, for example, if they're not careful, you know, if they didn't stay hydrated throughout the marathon, because you know they have the water stations. If you've ever watched a marathon or watched segments of a marathon, you know that they can get water as they go along. But if they wait and drink all their water at the end of the race, they can get very, very sick very quickly because they suddenly start putting this huge amount of water into their blood, which ends up around the cells, diluting the water in the interstitial fluid, causing the cells to expand rapidly. It also works the other way. A person who is dehydrated, severely dehydrated, when someone doesn't stay hydrated, the first place it affects is the fluid volume in the plasma. That plasma, though, is still leaking out into the interstitial spaces, but there's less water now. There's less water available to leak out into the interstitial spaces. The salt concentration, the amount of salt present hasn't changed. So it makes the water outside the cells saltier since there's less water there. It's like you take a beaker of water. I got a beaker containing a liter of water, and I put in 100 grams of salt. So I got a 10% salt solution. If I let it evaporate down to a half a liter, you know, I come back a week later and there's only 500 mils of water in here, it still has the same 100 grams of salt. It's twice as salty as it was before. Salt didn't go anywhere, the water did. So now in your interstitial fluid around your cells, that water, which was the same concentration as inside the cells, is now twice as salty. What's the cell going to do? It's going to send the water out. The cells are going to start shrinking. They don't like that either. Then they start dying too. So anyway, in our bodies, we pay very close attention to our water concentrations because osmosis isn't always going to do the right thing for us. It can make us move water from one side of a cell membrane to another if we inadvertently overhydrate or underhydrate. And you know, those kind of things can happen all the time. So, now, okay. So osmosis, again, is movement of water from the high water side to the low water side. High water and low water are determined by the solute concentrations. And the osmotic pressure is going to drive the water to go from its higher water side to its lower water side. Let's see what this shows us here.
Membrane is permeable to water molecules, and the movement of water into and out of cells is critical to life. Diffusion of water molecules across a selectively permeable membrane is a special kind of passive transport called osmosis. Plant cells are surrounded by rigid cell walls. When plant cells are exposed to hypotonic environments, water rushes into the cell, and the cell swells, but is kept from breaking by the rigid wall layer. The pressure of the cell pushing against the wall makes the cell turgid and is the desired state for most plant tissues. For instance, placing a wilted celery stalk or lettuce leaf in a hypotonic environment of pure water will often revive the leaf by inducing turgor in the plant cells. Animal cells lack rigid cell walls. When they are exposed to hypotonic environments, water rushes into the cell and the cell swells. Eventually, if water is not removed from the cell, the pressure will exceed the tensile strength of the cell and it will burst open or lyse. Many single-celled protists living in freshwater environments have contractile vacuoles that pump water back out of the cell in order to maintain osmotic equilibrium and avoid lysis. Plant cells are surrounded by rigid cell walls. When plant cells are exposed to hypertonic environments, water rushes out of the cell and the cell shrinks away from the rigid wall. These cells are dehydrated and lose most or all physiological functions while in the shriveled state. If cells are returned to isotonic or hypotonic environments, water re-enters the cell and normal functioning may be restored. Animal cells lack rigid cell walls. When they are exposed to hypertonic environments, Water rushes out of the cell, and the cell shrinks. The resulting cells are dehydrated and lose most or all physiological functions while in the shriveled state. If the cells are returned to isotonic or hypotonic environments, water re-enters the cell and normal functioning may be restored.
A good example of this is a red blood cell. You take red blood cells and put them under a microscope, and you add fresh water, distilled water, to the red blood cells, they will explode because there's a higher water concentration outside the cell, outside the red cell, if you add distilled water to it. And so the cell will let water come in, and it'll swell up and pop. And it usually happens so rapidly. You put a drop of distilled water on the slide, it sucks up under the cover slip, and the next thing you know, you have a red smear. And that's because the cell's just ruptured. On the other hand, if you take living red cells and put salt water on them, they will shrivel up as they pump all their water out that they have around the hemoglobin to try and reach equilibrium with the outside. And it's what we call crenation. They shrivel up, and if it doesn't get fixed, they're destroyed. Either way, too much water or too little water is a bad thing for us. Okay. And this is what happens here. We have three terms here. We have hypertonic, hypotonic, and isotonic. A hypertonic solution occurs when the the salt concentration on the outside of the cell is greater than what's inside. Hyper means great. So a hypertonic solution has more salt, more solute. It's usually sodium chloride anyway. So more salt on the outside, so water leaves the cell and it shrinks. A hypotonic solution, hypo means less, a hypotonic solution has less salt outside than inside the cell, and water goes into the cell and the cell will swell up. Neither one of those conditions are ideal for us. What we like to see is an isotonic solution where the concentration of the water outside the cell and the interstitial fluid is exactly the same as the water inside the cell. So there's water movement, water goes in, water comes out. We don't like to see our cells explode. We don't like to see them shrivel up like this. We see, the example for plants. You've all seen a plant that badly needs watering, and you add water to it and come back a few hours later and it looks fine. It was very limp because the cells have shrunk. The cell walls are holding everything intact. We don't have that luxury of having cell walls, which is probably a good thing. So anyway, the term here for these solutions is tonicity. Tonicity is what happens when you're exposed to different concentrations. As again, the isotonic solution has the same osmolarity, meaning the same, it's a nice way of saying the same concentration inside the cell as outside the cell. Water movement is in equilibrium. Nothing changes. Hypertonic solutions cause the cell to shrink because there's always less water outside than there is inside. So the cell pumps water out and it shrivels. And in a hypotonic solution, we have water coming into the cell and the cell can rupture. We call it lysing of cells, which is why you never give distilled water to a patient in an IV line. You would never administer an IV that is pure water because it would cause red blood cells to start popping. They'd rupture, they'd lyse. And of course, you'd lose all that hemoglobin. Now, sometimes, it's important, to and getting into the whole IV approach, when a person has been bleeding out and we don't have any blood available immediately, you can start an IV with what we call normal saline, 0.9% sodium chloride. It matches the salt concentration in blood. It's a volume expander. It fills up the space where the blood was, and that will keep the pressure from getting too low, and it'll give the heart something to pump. So we can raise our blood volume very quickly with these volume expanders, these fluid expanders, simply by using salt water of the same concentration as the plasma, normal saline. If your patient is dealing with edema, and there's a pathological state called congestive heart failure, where we have fluid accumulating in the lungs or fluid accumulating in our tissues, and your patient swells up. If you administer very carefully a hypertonic solution to your patient, it will cause that water to be drawn out of the cells, and they can then pee it away. There are numerous types of drugs that we call diuretics to help get rid of fluid accumulation in here. Hypotonic solutions, we never give those. That would be like distilled water, because that would destroy the red cells. So isotonic is done all the time. If you've ever been in the hospital and had an IV line running into you, just to keep the vein open, it's nearly always going to be normal saline. That's sort of a matter of routine. And then you can piggyback other medications onto that IV line. It's extremely unusual when you would have to administer a hypertonic solution to a patient, and you would never administer a hypotonic solution whatsoever. Okay, that's a great place to stop for today and for the week. Anyone have any questions on any of this? Believe me, it gets better. I'll just say that. Okay, so we will continue onward with this next week. Anticipate the lecture test either on ground on Thursday or opening up online on Thursday. So, and we'll just go from there. If you have any questions on any content, let me know. I'll be glad to answer them.